Hello everyone, I am Dr. Kavita Gajbhie, Assistant Professor in Pune College of Pharmacy. Today I am going to take subject or topic for the lecture is protein and peptide drug delivery system. So before starting protein and peptide drug delivery system, it is very necessary to understand what is actually the protein and what is the meaning of peptide. Protein are the large organic compounds made of amino acids arranged in a linear chain and joined together by peptide bonds. So here, in case of protein, the number of amino acids are more than 50. And if we talk about the peptides, so these are the short polymers formed from the linking in a defined order of amino acid. So here peptide in which the number of amino acids are present less than 50. So this is the major difference between these two type of moieties. Now if we see the structure of protein, so proteins are available in the four different kind of structure: primary, secondary, tertiary and quaternary structure. Primary structure, it is an array of number and specific sequence of amino acid in protein structure and it is available in a linear form. Secondary structure, regularly repeating local structure stabilized by the hydrogen bonding. Tertiary structure, three-dimensional structure of functional proteins are present. We can see the protein in a three-dimensional manner. Quaternary structure contains two or more polypeptide chains associated by non-covalent forces. Now the functions of protein. So protein play a very important role in our daily life. So here a few functions or few that point we can take, like transport and storage of small molecules, coordination or coordinated motion via muscles contraction, mechanical support from fibrous protein, generation and transmission of nerve impulse, enzymatic catalysis process, immune protection through antibodies, and also growth of control growth of and differentiation via hormones. So all these play very important role for functioning of our body. Now, what are the importance of protein? So few examples we can take. By this we can easily understand what are actual importance and applicability, importance and important functioning or important characteristic of protein in our body for functioning the proper way of our human being. So erythropoietin used for production of RBC, tissue plasminogen activator is used for treatment of heart attack, and oxytocin maintain the labor pain, bradykinin increase the peripheral circulation, somatostatin in decrease the bleeding in gastric ulcer, gonadotropin induce ovulation, insulin maintain sugar, blood sugar level also. So all these are the hormones which act as a controller or which act as a that kind of treatment therapy in our body. Basically, these are from origin, everything is our protein. So protein, as we all know, it is a macromolecule, and various kind of barriers are present in our body which restrict entry of protein or macromolecule in the internal part of our body. So more basically we can say four kind of barriers are present in our body. Enzymatic barrier limit absorption of protein drugs from GI tract. Intestinal epithelial barrier involved in the transport of protein drugs across the intestinal epithelium. Capillary endothelial barrier, which involved in transport of protein drug across the capillary endothelium. And very difficult or very complicated barrier is blood-brain barrier. The astrocytes and pericytes forms very tight junctioning over the blood capillaries of our brain. So it restrict or diminish or it totally retard the entry of not only protein peptide but other molecules also inside the brain area of our that human body. What are the pharmaceutical approaches of proteins? So chemical modification we can do. By this chemical modification, we can enhance the entry or enhance the approach of protein or macromolecule entry inside the body. Enzymes, protease inhibitors also we can associate with the proteinous formulation. Penetration enhancer also we can include. Formulation vehicles also we can apply. Mucoadhesive polymeric system also we can prepare, and also few pumps which deliver the protein in a specific or sustained manner. So first approach is chemical modification. That chemical modification which improves the enzymatic stability and membrane permeation property of the protein or peptide drugs. And it also minimizes the immunogenicity of the protein or peptide molecules. So product approach include amino acid modification. That modification we can do in the individual amino acid by the substitution of D amino acid and the L amino acid, and alter the physicochemical property of protein and peptides. For example, desmopressin and D-amino vasopressin, which is the conversion of D form to L form. Which also after that modification or alteration we can enhance the penetration capacity of protein inside the human body or through the barriers. Hydrophobization, the surface modification using the lipophilic moieties. So Nobex insulin is the best example of this hydrophobization technique. Now second approach is enzyme protease inhibitors. So the whole GIT and liver tend to metabolize protein and peptide into smaller fragments, or we can say where the number of amino acids only present 2 to 10, with the help of variety of proteolytic enzyme, because in our that esophagus, in our digestive tract or in our GIT tract, lots of proteolytic enzymes are present. So if we incorporate some type of protease inhibitors or co-administered with the protein and peptide, which alter the environment of minimum enzyme stability and suppress the proteolytic activity. So for example of some protease inhibitors, protease enzymes are pepsin, papain, thrombin, carboxypeptidase. Now, third approach is penetration enhancer. So if we incorporate some penetration enhancer in the formulation of protein and peptides, so that is important to disrupt the mucosal barrier to improve the permeation of large macromolecule substances like protein and peptide. So here few penetration enhancer we can incorporate for more or proper delivery of macromolecule through the barrier system. So for example, surfactant, polysorbate, SLS, chelating agents, EDTA, fatty acids, sodium caprate, mucoadhesive polymers, thiomers, cellulose derivatives, and phospholipids, phosphatidylcholine. And for this formulation approach vehicle, so the oral delivery of therapeutic protein and peptide can be successfully achieved by various kind of carrier system like dry emulsion, so prepared by drying the liquid oil in water emulsion containing a solid carrier in the aqueous phase, cellular carriers, for example, released erythrocytes, microspheres, liposomes, nanoparticles, and hydrogels. And also one more approach is mucoadhesive polymeric system. So these systems prevent the pre-systemic metabolism of the therapeutic protein and peptide, for example, thiomers and polyacrylic acid derivatives and cellulose derivatives. And our last approach is pumps. So pumps, a kind of device that we can attach or implant inside the body, so that can deliver the drug in a sustained manner, in a rate limiting manner, or for a longer period of time. So if we are talking about the pump, so here is very necessary to talk what are the category of pumps, like implantable pumps or mechanical pumps. So here implantable pumps, we can implant subcutaneously and delivered by IV infusion and are filled with the drug through a septum with a needle, and it delivered the drug to a certain vein for 7 to 14 days at a constant rate. And mechanical pumps are easily manipulated to deliver protein and peptide drugs. For example, it has been successfully delivered by portable syringe. Few marketed formulations are also available of protein and peptides, like Lupron, in which leuprolide used for prostatic cancer, Pergonal, FSH and LH used in case of female infertility, calcimer, solmone calcitonin drug used to treat hypercalcemia, and Ector corticotropin, it is used in case of hormone deficiency. And now if we talk about the recent advances, so what kind of recent advances we can associate with the already available approaches? So here we can incorporate some smart type of polymer which can deliver the drug in a specific stimulus condition, like in case of pH, in case of thermal condition, in case of any redox condition, in case of any enzyme or biological fluid is present in a specific area of our body, so that can get a kind of trigger or stimulus, so deliver the drug in a proper manner. For example, temperature sensitive polymers or polypeptides we can use, that proteins also we can use, like polyethylene oxide, polypropylene oxide, polyethylene oxide, triblock copolymers, and phase sensitive polymers like polydiol lactide, polydiol lactide coglycolide, pH sensitive polymers, polymethacrylic acid, and G-ethylene glycol, photosensitive polymer, polyvinyl alcohol. So these kind of polymers also we can incorporate for modification and alteration of delivery of protein and peptide. So alter the that environment and alter the release of drug in a specific stimulus condition. Hybrid protein delivery also. So heterogeneous hybrid proteins can be designed wherein the combined or recorded feature of one or more protein that display effector function, protection abilities, and recognition properties. Site-specific hybrid protein may be produced, ligated gene fusion hybrid delivery system, and synthetically linked hybrid conjugate. So these two approaches are very much popular nowadays in modern era for delivery of drug, that type of macromolecules. Thank you so much.